Timing and number. Background. A popular tale recounts the story of a hunter who wished to shoot a crow, but the bird repeatedly eluded him by flying just out of range whenever he appeared. Hoping to deceive the crow, the hunter asked a friend to go with him behind a hunting blind and then leave after a short time. Surely, the hunter thought, if one man left, the crow would not realize that one remained and would return to the area. Yet, the bird was not fooled by this ruse, or another attempt in which 3 men entered and 2 emerged. Only if 5 men entered the blind was the crow's ability to keep track of the hunters exceeded. Credit for the original report of the hunter's story is often given to Sir John Lubbock, though Ayn Rand's mention of the story in Introduction to Objectivist Epistemology, Rand and Peikoff, 1979, helped to spread the tale. If true, the story would suggest that even without a formalized symbol system for counting, e.g. the use of Arabic numerals, a bird species can track at least 1 to 4 potential predators, represent this quantity while the predators are out of sight, and then update this representation if the predators are seen to leave. It would also suggest, though, that there is a limit to how many hiding individuals can be precisely represented. Anecdotes such as this, of course, do not provide the basis for good scientific study, yet, whether or not the story is true, it raises questions that comparative cognition researchers, often alongside developmental and cognitive psychologists, have been working to answer for many years. Along the way, many commonalities have been found between how the animal mind represents number and time, and studies of one have often shed light on the other. Numerical cognition and timing both involve discriminating magnitude, and there is now good reason to think that some neural systems may be shared by these 2 processes. Additionally, both seem to be present across many animal species and emerge very early in human development. Indeed, given what is now known, the hunter's story is not far-fetched. Chapter Plan. This chapter begins with a description of animals' ability to keep track of time, both for long durations such as days and short intervals of the order of minutes. The study of interval timing connects closely with the study of numerical processing. Research has repeatedly pointed to similar or shared cognitive systems that allow for interval timing and approximating number. After discussing the approximate number system, a second system for small quantities will be detailed, followed by a consideration of operations such as addition, subtraction, and ordering. Sensitivity to time and number is likely adaptive for many behaviors, including foraging. Timing may provide information as to the availability of food sources that replenish over delays, and numerical discrimination can provide a basis for choosing foraging sites. The importance of these abilities suggests that one might expect to see evidence for them across a wide range of species, and as will be seen, the study of timing and number has benefited greatly from a comparative approach. Periodic timing. It is relatively easy to observe instances in which animal behavior is influenced by time. Many pet owners know the joy of being awakened by a restless dog that wants to walk early on a Sunday morning, and while on that walk, it is common to witness that the early bird truly does get the worm. 2 primary types of timing have been characterized, one for cyclical activity that occurs during a 24-hour period, which will be referred to as periodic timing, and one timing system for much shorter durations on the order of seconds or minutes, interval timing. The latter will be presented in section 6.2. Almost every organism that has been studied shows a rhythm of activity or physiological state that matches the day-night cycle of Earth. Importantly, though, this rhythm can persist even when organisms are moved to consistent light or darkness. The circadian rhythm appears to be the result of an internal clock with an approximately 24-hour cycle that can undergo entrainment by or fall into synchrony with the external day-night cycle. For example, in an experiment with cockroaches that were normally active at dusk, covering the insect's eyes did not stop the rhythm of daily activity. However, Without the light-dark input from the environment, the rhythm followed a slightly shorter period. Activity rose every 23.5 hours, meaning that over time, activity occurred earlier and earlier before dusk. This free-running rhythm is thus only close to 24 hours. It is circa near a day. When sight was restored by uncovering the eyes, the cockroach's internal rhythm slowly became synced with, entrained to, the natural day-night cycle again. Across species, there appears to be an independent internal clock that can be modified in potentially adaptive ways by the environment to influence behavior. Time and place learning. The internal circadian rhythm is typically discussed in terms of physiology, not cognition. However, animals may learn about events that occur at certain times of day, linking the event to the internal clock. For some carnivores, for example, the emergence of prey may influence daily activity routines, thus optimizing hunting efforts. Many flowers produce nectar at certain times of day, thus bees and hummingbirds may effectively feed through time and place learning. Inspired by these observations in the wild, researchers have systematically examined time and place learning in the laboratory. In one classic example, Bieback et al. 1989 housed garden warblers in an enclosure consisting of a central room surrounded by 4 feeding rooms, figure 6.1. Unlocked food containers were only available in one feeding room at a given time, corresponding to each 3-hour block of a 12-hour light cycle, i.e., Room 1 had food available from 900 to 1200 hours, Room 2 from 1200 to 1500 hours, etc. If a bird entered a room at the correct feeding time, it could eat from the unlocked container for 20 seconds. Over 10 days, the birds learned to enter rooms during the appropriate 3-hour block. In subsequent testing, each of the 4 rooms had food containers that were unlocked throughout the 12-hour light cycle. Although birds could now eat in any of the rooms at any time of day, they continued to visit the rooms based on the original schedule. In a second experiment, birds were prohibited entry into any of the rooms during one 3-hour block. When entry was again permitted during another block, the birds reliably entered the appropriate room for that block, suggesting that performance was not simply based on a learned pattern of activity alone, e.g., enter room 1, then room 2, etc., but was due to the linking of the food locations with the circadian rhythm. Figure 6.1, Time and Place Learning in Garden Warblers. In the training phase, middle graph, food was only accessible in a feeder in one room for every 3-hour block. During the test phase, bottom graph, the warblers continued to search for food in relation to the time of day, even though all rooms were accessible. Okay, so at the top you can see a diagram just showing all of the rooms and that they're connected to each other. So in the first graph you can see on the y-axis is number of choices from 0 to 6, and on the x-axis is time of day in hours. So it's divided into those 4 3-hour periods of 6 to 9, 9 to 12, 12 to 1500 hours, and 1500 to 1800 hours. Okay, so you can see in the first chunk, they go to the first room. Second chunk of time, they go to the second. Third, they go to the third room. Fourth, they go to the fourth room. Okay, and then the bottom graph is the test phase, and it still shows the same pattern. The first going to the first room, second block going to the second room, third block of time going to the third room, and fourth block of time going to the fourth room, even though they could have gone to any room because the rooms were all open. So clearly the birds are keeping track of this because it's not restrained. But then, of course, they also tested by modifying some things that the birds aren't just like following a script of which room to go to either. By blocking all the rooms, then the birds still kept track of time and went to like, if block 2 of time was like blocked, all the rooms are blocked, then like when it was unblocked in block 3, the birds would appropriately go to block 3 in the room 3 in the third block of time. So birds are still keeping track of time and not just following the pattern because the pattern was disrupted. They're following the timing. Mechanisms of time and place learning. For the garden warblers in the previous example, it is likely that the state of the internal circadian rhythm acted as a contextual stimulus to which the correct food location was associated. Additionally, the circadian clock itself was likely entrained to the light-dark cycle. Evidence for this comes from an experiment in which the lights remained on for 24 hours. Birds still visited the rooms during their specific 3-hour block, at least during the first day of testing. After that, the visits to feeding rooms began to diverge from the 24-hour clock, instead appearing to follow the free-running rhythm. In mammals, the light-entrained circadian clock is thought to be located in the suprachiasmatic nucleus, SCN, of the hypothalamus. The eyes provide information to the SCN about illumination through the photopigment melanopsin, which is sent from retinal ganglion cells to the SCN. The SCN can then pass on information to the pineal gland, which in turn secretes the hormone melatonin. In humans, the circadian rhythm can be entrained by manipulating the ambient light to shorter and longer periods than the Earth's 24 hours, though the differences were slight, 23.5 hours and 24.65 hours. Timing intervals. In periodic timing, an internal clock can be entrained to important environmental stimuli, such as the day-night cycle, in turn influencing behavior. In interval timing, however, behavior can be influenced by relatively arbitrary, short durations that are signaled by relatively arbitrary stimuli that can occur at any time of day. The importance of short durations on learning was discussed in Chapter 4 in relation to temporal contiguity of stimuli. Associations between two stimuli are formed more easily if they occur close together in time. The work presented here focuses instead on the ability to time short durations and discriminate between them. Much of this work has been done in the laboratory, where experimenters create the relevant intervals, though timing of naturally occurring intervals likely influences behavior in the wild. Laboratory procedures. In 1981, Roberts developed a new procedure for examining timing in animals, the peak procedure, which was actually based on previous work by Catania, 1970, involved exposing rats to many trials in which food could be accessed a fixed time after the onset of a stimulus, e.g. a light. When a light came on, it signaled that food could be obtained by pressing a lever 20 seconds later. After food was obtained, the light turned off in preparation of the next trial. Occasionally, empty trials were included in which food was not delivered upon the first lever press at 20 seconds, and instead, the trial ended when the light turned off much later. So this is operant conditioning. Recording the rate of lever presses on these trials reveals a rather unsurprising finding. The rats do not press the lever only at exactly 20 seconds. They are hungry and tend to press multiple times, both before and after 20 seconds, on those empty trials. Indeed, without consciously counting seconds, adult humans would have difficulty being exact in their responses. But two aspects of the behavior are particularly noteworthy. First, the rats' responses peak very close to 20 seconds and follow a relatively normal distribution. The timing is quite accurate. Figure 6.2. Second, if a stimulus signaled that food could be obtained after 40 seconds, responses again peaked at 40 seconds and followed a normal distribution, but now, the spread of the curve, the variability, was much greater. In fact, the variability of the curves is proportional to the length of the interval being timed, which has been termed the scalar property. Figure 6.2. Example peak procedure data from Roberts, 1981, for rats trained with reward for lever press at 20 seconds, seconds 20, and 40 seconds, seconds 40. Roberts details the statistical methods of fitting the curves in detail. So on the y-axis is rate, the response per minute of lever presses from 0 to 60, and on the x-axis is the time in seconds from 0 to 80 seconds. Okay, so on the first curve, it peaks at 20 seconds, and the second curve peaks at 40 seconds, so that must be the two conditions that they've been talking about. And at 20 seconds, it is a narrower normal curve, showing that there is less variability around the 20-second mark, and it is much broader for the 40-second mean curve, showing there is much more variability because the time interval has increased by 50%. The general finding from the peak procedure that responses to longer time intervals occur with greater variability fits with Weber's law, which was introduced in Chapter 2. Other experimental procedures produce similar results. Church and Gibbon, for example, presented rats with certain intervals that would be reinforced, while other durations yielded no reinforcement. The rats were placed in a lighted chamber, but the lights would turn off for intervals that ranged from 0.8 to 7.2 seconds. When the lights were turned back on, lever pressing would be rewarded if the darkness interval had been 4 seconds. After extensive experience, rats' lever pressing behavior was highest if the darkness lasted exactly 4 seconds, and the behavior decreased as the interval duration differed from 4 seconds. That is, temporal generalization occurred. The responses continued for durations that were different yet close to the target duration. Further, analogous to the results from the peak procedure, the generalization gradients for target durations longer than 4 seconds had greater variability. Although the majority of experimental evidence comes from rats and pigeons, interval timing appears to be present across many vertebrates and invertebrates. Bumblebees, for example, have been tested using fixed interval procedures in which a reward is received on the first response that occurs after a fixed interval of time has elapsed. The response in question was the extension of the proboscis, which naturally occurs during feeding. A bee could obtain sucrose by extending its proboscis through a small hole in the experimental chamber. During a trial, a chamber light was illuminated, and the interval, e.g. 6 seconds, began. The first proboscis extension that occurred after the interval elapsed yielded a sucrose reward. Typically, vertebrates tested using these procedures withhold responses until later in the duration, and the highest probability of responding occurs at the end of the interval. Similarly, bumblebees responded maximally at the end of the interval. Timing in the wild. It is possible that, in the wild, the timing ability of bumblebees supports their feeding behavior. Pollinating species often show resource fidelity. They repeatedly visit the same nectar sources. Food sources such as flowers do replenish their nectar after a pollinator has fed, but this process takes time. To feed efficiently, then, pollinators should schedule visits to food sources according to temporal schedules. What this would require is remembering not only where the feeding site was, but also how much time has passed since the last visit. As mentioned in Chapter 5, rufous hummingbirds can use landmarks to remember locations and avoid recently depleted flowers. Similar experimental techniques can be used to examine whether and how these birds keep track of the time interval between visits. In one such study, wild, territorial male rufous hummingbirds living in the Rocky Mountains of Alberta, Canada, were tested using arrays of 8 artificial flowers that could contain a sucrose solution. In each bird's territory, experimenters refilled 4 flowers 10 minutes after the male fed and emptied them. The other 4 flowers were refilled 20 minutes after the bird fed. Both of these durations are shorter than the typical nectar refilling rate of flowers, but allowed the experimenters to observe the hummingbird's behavior. After experience with these refill rates over many days, The birds were revisiting the flowers that replenished after 10 minutes sooner than they revisited those that replenished after 20 minutes. Further, the visits appeared to match the refill schedules, figure 6.3. Similar to the laboratory studies of interval timing, the visits peaked at the appropriate times, but there was variance. While it was not the case that this variance was truly proportional to the refill durations, there was greater variance for the larger 20-minute duration. Figure 6.3. Example artificial flower and data from Henderson et al. 2006. The graphs show that the birds visited the artificial flowers at intervals consistent with the refill rate, 10 or 20 minutes, with greater variance for the 20-minute duration. Okay, so there's a series of graphs. On the y-axis is the post-reinforcement pauses, PRPs, from 0 to 600, and on the x-axis is time in 5-minute bins. Okay, so each graph is tracking one unique bird in their post-reinforcement pauses. Okay, so for the flowers that had a 10-minute refill time, yeah, they did actually visit them around 10 minutes, and it is almost a normal curve. And for the 20 minutes, it did peak around 20 minutes. It's not a normal distribution. It's not that symmetrical. They visited still generally earlier than that, but that might have been skewed by visiting the 10-minute ones. And then, yep, that pattern, 10 and 20-minute mean revisitation is holding steady for, both of the two other birds. The rufous hummingbirds seem to have learned that the time until refilling differed among the flowers, and more specifically, they learned that the refill times were 10 and 20 minutes. Another way to consider this finding is the following. The birds remembered the refilling times for each of the 8 flowers and then updated this information across the many visits to the flowers throughout the day. Although there are not many studies of interval timing in the wild, it is possible that procedures such as these may complement and confirm results from the laboratory. The natural feeding behavior of pollinators is a likely situation in which interval timing may be observed, yet other situations in which animals utilize food sources that can be depleted and then renewed might also indicate the timing of intervals, e.g., the feeding behavior of cleaner fish that remove ectoparasites from other fish species.